So I'm working at the University of Geneva, so division of child development, and I'm based in a laboratory which is in Lausanne, EPFL, which is the CIBM, so Center for Biomedical Imaging, where we are performing imaging on preclinical scanners, so with small bore and mostly on rodents, and MRI and MRS, and mainly diffusion imaging. So I'm going to talk today about application of MRI and MRS, so imaging and spectroscopy, to animal models of perinatal brain injury. Okay, so when we talk about perinatal brain injuries, we talk about brain injuries occurring during the perinatal period, meaning between 32 weeks of gestation to 7 days postnatal, and with consequences as lifelong neurological disabilities. There is a global concept that has been proposed by Joseph Volpe, which is the dad of neonatology, which is called the encephalopathy of prematurity for babies' births before 32 weeks of gestation. And this encephalopathy of prematurity is characterized by white matter damage, but also an altered cortical development. And for these babies with a born weighting under 1.5 kilogram, we will have 25 to 15% with developmental disabilities and 5 to 15% with more major cerebral palsy. The etiology of these injuries is complex, and most of the time it's multifactorial, but we know that the main causes are hypoxia ischemia and or infection inflammation. And as such, some hypoxic ischemic or inflammatory animal models have been developed. So for animal models, we need to choose also which kind of spaces we want to use. So we have the rodents, so the mice or the rats, the piglets, the sheep. So when we look at these pigs, these guys are really different from this one. But if we go a bit deeper, so the rodent's brain, they are non-girified, but if you look the piglet or sheep brain, they are girified, and finally they are quite close to the human brain. So depending on what you want to do, rodents are very useful if you want to study the time course of the development. The gestation is very short, it's 20 days, and you can assess the cerebral development from preterm to adulthood in almost 2 months. A rat pup at postnatal day 3 roughly corresponds in terms of cortical development to a baby birth at 24, 28 weeks of gestation. So postnatal day 7 corresponds to the human term, and from postnatal day 10 to 44, you have the myelination process. The setup is very easy, it's accessible, it's cheap. You have plenty of knockout models, or you can create your own models, hypoxic, ischemic, inflammatory models. You can perform in vivo, longitudinal, multimodal MR studies. It's what we do on our preclinical scanners. Major drawback is the white matter gray matter ratio that is really different than what we find in human. There's not a lot of white matter, there's a lot of gray matter. And the brain is non-girified, so you will have some limitation for the models. And if you want something closer to human, you need to go to the girified brain, so sheep or piglet. Our gestation is a bit longer, but you have a similar white matter gray matter ratio and a girified cortex, so you are closer to the human brain, and the lesion, HI or inflammatory induced lesion, will be also closer to what we see clinically in baby lesions. You can also perform the operation in utero on the fetuses, hypoxic, ischemic, or inflammatory. and in terms of ethics, it's also easier than using primates. But setup is more complicated than for rodents, and it's also more expensive. And for us on our scanners, we can perform only ex vivo MR studies because we have small bore, so we cannot put a sheep or a piglet into the scanner. So we will have anatomical, structural information, but nothing about the metabolism, for instance. I say most of the time because we're also working in European project with a team in Maastricht, where they are doing in vivo MRI on sheep brain. I will show you some images. Okay. So diffusion tensor imaging, I don't know if you know or not this technique, just it's a technique that has been widely used to assess the healthy and pathologic brain development. It corresponds to a 3D measurement of water diffusion due to thermal agitation in the tissue. So due to thermal agitation, the molecule bumps and they diffuse in the tissue. And when you acquire this or measure this diffusion tensor, you can derive from the tensor some interesting quantitative parameters, which are the diffusivity values. So the mean diffusivity, which corresponds to the ADC that was exposed before. You have the axial diffusivity, which is the diffusivity value along the principal diffusion direction. The radial diffusivity, which is perpendicular to the principal diffusion direction. And the fractional anisotropy, which roughly corresponds to the deviation between these axial and radial diffusivity. So if you are in a cellular structure, for instance, you see these axial and radial diffusivities that are very close, and the fractional anisotropy is very low. If you increase the cellularity, you will have your axial and radial diffusivities that will be reduced, but the FA will not change because they are close again. If you are in a white matter structure, so you have these fibers, it's like if you have some hose pipes, for instance. So you see that the diffusivity, you have one principal diffusion direction along the fibers, and the radial is shorter. And if you increase the fiber density, for instance, you will reduce this radial diffusivity, and the fractional anisotropy is going to increase. So this technique has been used already to assess the cerebral development in rodents, in humans, on sheep, in gray matter and white matter. So mainly, if you look in the gray matter, so in the cortex, with the development, cerebral development, you have a decrease of this anisotropy, mainly due to the neocortical maturation and the dendritic arborization. So you go from a radial organization of the tissue to kind of branches with the dendrites. And in the white matter, you have an increase of the fractional anisotropy attributed to the pre-myelinating setup first, then the myelination, because before the beginning of myelination, you have an increase of the fractional anisotropy. So during the white matter development, you have the oligodendrocytes that are situated between the myelinating fibers, and they are creating the myelin sheath by wrapping around the axons. And these oligodendrocytes, they are differentiated between 28 and 40 weeks of gestation. And during this differentiation, they show some different characteristic, and actually when they are changing from pre-oligodendrocyte to pre-myelinating and myelinating oligodendrocyte, we have this fractional anisotropy, which is increasing because of the microstructure which is changing. But depending on the time point of the insult, you will not have the same vulnerability of this oligodendrocyte, so you will not have the same kind of lesion. Even if you are working on animal models, you need to choose when you want to do your inflammation or hypoxia ischemia to be sure that you will have the kind of lesion you desire. Uh, These oligodendrocytes, they also have a trophic role for the axonal development, and it's why we have in these kind of brain injuries also a cortical impairment by the projection fibers, the periventricular white matter too, and from the cortex. So when you want to do diffusion imaging, it's like when you are going to a restaurant, depending on your appetite, on your money, on your time. So you can do the basic, or the basic is just the ADC, so diffusion weighted images, but if you want the tensor, the basic is the DTI with 6 directions, so it's the minimum you can acquire to have access to these parameters, so diffusivity values, mean diffusivity, axial diffusivity, radial diffusivity, fractional anisotropy maps, and direction encoded color maps. So it's the same as this one, but the direction of the fiber is color coded. The red is right left, the green is up down, and the blue is antero posterior. If you if you have a bit more money and bit more time, you can take the DTI 40 directions. So mostly you will increase the accuracy of your measurement, but it will be a bit more time consuming. You will have access to the same kind of maps, same kind of images, but with better accuracy. On the rodents, it's not really necessary, I would say, because the brain is not too complex, but it has been shown on humans that to have an accurate measurement of fractional anisotropy, you need at least 20 different directions because the brain is more heterogeneous. Okay. So DTI is very useful, and if you look just on PubMed, just typing diffusion tensor imaging, you see that in 2023 we have more than 23,000 publications. Why? Because the DTI derived indices, so these diffusivity values and the fractional anisotropy, they are really, really very, very sensitive to the tissue microstructures. It means that you can characterize plenty of pathologies that affect the brain microstructure. With DTI, you will see something, you will be able to see something. But the problem is that it's very, very sensitive, but not enough tissue specific. It means that, for instance, you can have an FA decrease with a myelination defect, with an axonal diameter increase, with diffuse axonal injuries, with interaxonal edema. And something which is important in preclinical imaging, but also in clinical imaging, is try to find something which is more specific to be able to understand the mechanisms of the lesion or of the development. So for this, there are some models that are under development for many years already, but which we call the multi-compartment models. So if we go back to our restaurant, and if you want to increase the accuracy of your measurement, but also increase the specificity of your measurement, you can go, for instance, to NODI, which is the neurite orientation, dispersion, and density imaging. So you will need more direction, 96 directions, or 96 diffusion-weighted images in different directions of the space. So it's more expensive and more time-consuming. The idea of these multi-compartment models is to separate the signal from the different compartments in the voxel. Most of the time, and it's the case for NODI, you have 3 different compartments. So one which is the CSF, characterized by a free diffusion, so very fast diffusivity and anisotropy close to 0. The extraneurite compartment, which is characterized by an hindered diffusion with fast diffusivity and low anisotropy. and the intraneurite compartment, which is characterized by a restricted diffusion, meaning a slow diffusivity when you are perpendicular to the fibers, and high anisotropy. And with this model, when you fit your data, you have access to the DTI maps, but also the intracellular volume fraction or intraneurite volume fraction or neurite density index. There are several names for the same, which is the fraction of tissues that comprises neurites. So when we say neurites, it's neurons or dendrites. Isotropic volume fraction, which is the fraction of free water. The orientation dispersion index, which is an index that mimics the fanning of the fibers. And if you have more time, this morning in about the clinical scan, Stefan was talking about the acquisition time. If you have really long time, you can go to ex vivo diffusion MRI. So we can let the rodents under anesthesia 2 hours in the magnet without any problem, and we don't have a lot of problem of motion. But we cannot let the rodents during 20 hours, 24 hours. With ex vivo diffusion, you can let your sample during 24 hours in the magnet. So you will increase the SNR, you will have more accumulation, you can increase also your resolution of your images. You can use diffusion B values that are more important. So when you increase your B value, you decrease the signal to noise ratio. So you need to have a lot of accumulation to keep a good signal to noise ratio. But you will also improve the microstructure characterization. And you can also have a direct comparison with your histological data, which can be interesting if you want to validate your model in terms of methodology. So NODI is one of the model, and one of the model we use because we had a collaboration with Gary Zhang, which is the NODI guy in Geneva, but there are other models that we can see. There's a spherical mean technique where you have access to the diffusivity value inside the axons and the extra perpendicular to the axons, and the white matter tract integrity, which is a bit more demanding, this one, but you have roughly the parameter of this one and this one, because you have also this angle which correspond roughly to the ODI. What is important to keep in mind when we talk about these multi-compartment models, it's that in multi-compartment models, there is the word models, and it's really a modelization. It means that you will not have access to the absolute quantification of the intra-axonal volume, for instance, but just an intra-axonal volume fraction, meaning in your voxel, which part of the voxel, each intra-axonal, which part is extra-axonal, which part is isotropic. So it's not something absolute. Okay. Another way to increase the specificity of your measurement is the use of multimodality, meaning imaging and spectroscopy, which is actually not so easy because in terms of hardware, it's not always the same requirement. But we have the chance to have in Lausanne ultra high magnetic field magnet, so 9.4 Tesla and 14 Tesla. And with this, we have an increase of the spectral resolution and of the signal to noise ratio also. So we are able to have very, very nice spectra here in the cortex of the rat, where we have access roughly to 20 metabolites that we can absolutely quantify. And in these metabolites, you have neurotransmitters, neuronal markers, tissue integrity markers, energetic metabolism markers, membrane precursor, osmoregulation, myelination, glial markers, neuroprotection. So plenty of neurochemical information that are very interesting when you are working on the animal models. Sorry. So, for instance, for example, of this multimodality, we have this is a study on hypoxia ischemia on the rat at postnatal day 3. So they had a right carotid artery cauterization, followed by 30 minutes at 6% oxygen. And we looked the effect at P4, P11, and P25, so longitudinally on the same animals. So already at P3, we see on the T2 weighted images that we have an hypersignal in the cortex. So just 2 or 3 hours after the lesion, and at 24 hours, we see here that we have the size of the lesion, which is a bit bigger. So we were able to quantify the size of this lesion, so the percentage of injured cortex at 24 hours after the lesion. Then at P11 and P55, so 1 week and 3 weeks after, we looked at the percentage of cortical loss. So following the lesion, you have a cortical loss. We did a correlation between the size of the lesion at P3 and P4 and the percentage of cortical loss at P25. And we also had different responses to the same initial injury for males and females, with females that are less injured than males. So we did also spectroscopy on these guys in control group and in the lesion, in the cortical lesion. And when we look at the spectroscopy results, we see that in the acute phase, so at 24 hours after the lesion, we have almost all the metabolites that are decreased, except the lactate that is increased 24 hours after the lesion. And when we look at the long term, we see almost a large part of the metabolites that are restored, mainly due to the defense and repair mechanism that we have during the cerebral development. We'll also use this technique to assess the effect of lactoferrin as neuroprotective strategies on inflammatory models. So it's also a rat brain where we do intracarosal injection of LPS or NaCl. And we use lactoferrin, which is an iron-binding glycoprotein, which has several interesting properties. Antioxidant, anti-inflammatory, and antimicrobial. And the idea was to supplement the dams with lactoferrin during lactation, and that the pups receive the lactoferrin through the breastfeeding. So we looked with histology the inflammatory markers, and there was no effect of the lactoferrin on these markers following the inflammation. But we show with the spectroscopy in the striatum 24 hours after the injury that we had what we call the antioxidant profile. So it's the ascorbate and glutathione, where we had the concentration of these metabolites that were reversed by the effect of lactoferrin. We also performed some just T2 weighted images to quantify the size of the ventricle. So we have ventriculomegaly that was reduced by the effect of lactoferrin, and also with NODI we looked at the microstructures, and we see in the different white matter, gray matter region that we have some detailed parameters that were restored with the effect of lactoferrin. So with these study, we show first that we have an effect of neuroprotection with lactoferrin, and we have some mechanistic information on the path of the neuroprotection with this multimodality MRS and MRI. So the idea of this study is translation to humans. So the idea would be to give lactoferrin clinically as preventive treatment to mothers with premature labor and risk of preterm delivery, and then after delivery as a supplement for newborn neuroprotection. This is another study of mouse inflammation. So we assess by DTI. So it's really a characterization. It's a mild model. So they were IP injected from P1 to P5 with an interleukin or a saline solution. And we looked in different white matter and gray matter region, and we saw some changes in all the region we assessed, with mainly a decrease of the fractional anisotropy in the white matter and increase of the diffusivity value in the gray matter, so really diffuse diffusion changes. And these diffuse diffusion changes, they match very well with a diffuse reduction of the myelin protein that has been measured on histology by MBP and PLP. So a diffuse myelination defect, and we also saw with electron microscopy an oligopathy and axonopathy. So this study shows first that moderate systemic inflammation at a specific time during the development will alter the developmental programs with myelination deficits, but also we are able to characterize these lesions with diffusion tensor imaging. So yes, I said we did mainly ex vivo MRI when we are working on chips. So we have a collaboration. It's a European project, H2020, which is called PremStem, to try to use stem cells in perinatal brain injuries. And in Maastricht, they put the chip into a clinical scanner. So we are working on the images, and we were able to reconstruct the images with DTI and NODI and the spherical mean technique. So I just show the images. We don't have enough resolution to work on the cortex, for instance, but on the white matter tracts, we can see some interesting stuff which has not yet published, so I will not show you the result, but it was just to show the images. Okay, I will finish about NODI specificity. So I said before that the problem of DTI is it's not enough specific to the tissue microstructure, and that multi-compartment models could be an answer of this problem. So we wanted to see if it works or not. So we decided to examine the association between NODI and DTI derived parameter with histology, and we looked at in the developing cerebral cortex of the neonatal rat. Idea was to associate the neuronal dendritic complexity with this orientation dispersion index, which is supposed to mimic the fanning of the fibers, and the cellular process density with the neurid density index, or intracellular volume fraction, or intraneurite volume fraction. So we have some rat brain that have been scanned ex vivo, postnatal day 1, postnatal day 1, 3, 7, 14, 21, and 35. So we have the DTI derived parameters and the NODI derived parameters. We see on the images that we have really two different kind of contrast between postnatal day 1 and 7 and 14 and 35. It's really different. On the fractional anisotropy, intraneurite volume fraction, or orientation dispersion index. And if we look the quantitative values, when we look the fractional anisotropy, so we have a decrease from postnatal day 1 to postnatal day 7. And when we look now the orientation dispersion index, we see that we have an increase of this parameter from postnatal day 1 to postnatal day 14, and this match pretty well with the number of dendritic branches, which is increasing, and also the dendritic morphology we see here with the branches, the morphology which is really changing. If we quantify the neuronal dendritic density, so really the neurons, we see that we have a small decrease, then an increase. And when we look at the neurid density index, we have exactly the same path with a small decrease and an increase from postnatal day 1 to 3 to 7. And we have also a good match in this way. So about the specificity of NODI, we can say that NODI and DTI have similar sensitivity to the cellular changes in the cortical development with fractional anisotropy and orientation dispersion index that reflects the dendritic growth. But we have a better specificity of NODI to the cortical tissue microstructure with ODIs that reflects the neuronal dendritogenesis and the neurid density index that reflects the neuronal dendritic density. Okay, just to conclude, so I hope I convince you about the high relevance of animal models of perinatal brain injuries, of the use of multimodality, so imaging and spectroscopy, of the use of diffusion tensor imaging, which is really a good technique if you want to probe the microstructure, the brain microstructure. But we also need more specificity to understand the lesion neuroprotective mechanisms and these non-invasively, and that multi-compartment models can be an answer, but there are still a lot of work. Just want to acknowledge my colleagues from the different universities and thank you for your attention.